We learn about the replication of DNA in this session. And the process of replication is slightly different in prokaryotes and eukaryotes. But for the sake of simplicity, we'll understand the process of DNA replication in prokaryotes, and the prokaryote that we have chosen for our study is E. coli. Now, there are slight differences between replication in prokaryotes and eukaryotes as spoken before, but more or less the process remains the same, and that's why this process is basically a journal introduction. This presentation is introduction to the journal introduction to the process of DNA replication. Now, the process of DNA replication takes place in three stages: the stage of initiation or the beginning, the stage of elongation when the chain grows, and the last step is the termination, the process of termination of DNA replication when the replication stops. Now, the genome of the E. coli or the DNA of the E. coli, it's a bacillus. Okay, so this bacteria is a bacillus. It has got its DNA. It's fairly coiled DNA present in a certain region which is called the nucleoid. And if we take this DNA outside and we spread it around, we'll find that the two ends of this DNA are connected to each other. They are not so roughly connected, but very smoothly connected to each other. And because it's a closed structure, we can consider it as a circular genome. You should bear in mind that we don't find circles like these, but it's a closed structure, and we consider the genome of E. coli as a circular genome. Now, this particular genome has wide variety of sequences, a large number of base pairs which will act as genes. So we'll have a certain gene here, maybe we'll have a certain gene here, maybe we'll have a couple of genes here. So these particular segments called genes will produce certain proteins, right? Functional proteins or structural proteins, whatever. But for the sake of replication, because we are only interested in replication in this session, there are two regions that are of greatest importance to us. The first region on the genome is called the ORIC, or the origin of replication, and the other region is called TER region, or the termination region. Now, when the process of replication has to start, it cannot start randomly at any particular point. The proteins, they have to find a particular point from where they can start the process. This process of replication has a specific point where it starts, and that point is called ORIC, or the origin of replication. Now, the proteins or the enzymes that are involved in replication of DNA identify ORIC by its particular base pairing sequence. So this particular site ORIC has a specific set of bases. Okay, so there is this particular sequence G, A, T, C, T, blah 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 blah, and so on and so forth. So there are these sequences which are identified by the proteins and enzymes to start the process of replication. Similarly, if the process of replication has started, it will have to end at some time and at some point. And so there is this particular region. Now the bases here, the sequence of bases, the sequence of nitrogenous bases in the termination region are read by the proteins and functional proteins also, by which I mean the enzymes, will also read these particular base pair sequences as the end of the DNA replication. Now let us proceed further and look at each of these three stages in slightly greater details. Now first of all, let us go to initiation. Now initiation is basically the beginning of replication, and where does it start? It starts at ORIC in the circular genome. So we got the circular genome here, and probably here we got this particular site called the ORIC, origin of replication. Now what else is required for the process of initiation? There are certain proteins that are needed, particularly there's this protein called DNA which is required. Then we have a certain enzyme, which is, enzymes are basically proteins, okay, functional proteins or catalytic proteins. So an enzyme called DNA helicase is needed. Then we require certain more other types of protein which are called Ssb protein or single strand binding proteins. And we must bear in mind that the process of initiation, or rather broadly speaking, all the three stages, the initiation, elongation and termination, all the three processes or these three stages require a lot of energy. They are energy driven processes, and that's why they use up energy present in molecules like ATP. But ATP, other than ATP, large number of other molecules are used, which we'll see in just a little while. So far we've done what are the requirements for initiation. Now how does this initiation take place? So what we have here is, let us just take this particular segment ORIC. So ORIC will have like two strands, right? And we'll have base pairs in it. And so this particular segment, which is called the ORIC or the origin of replication right here, is first of all identified by a special set of proteins. So a special set of proteins called DNA DNA proteins first of all identify this particular site, and once this particular site has been identified and marked that this is the OREC, these particular proteins called DNA proteins, what do? They recruit, or they bring together, or they signal this particular enzyme called DNA helicase also to come along and attach at this particular point. So this particular protein, functional protein called DNA helicase, with the help of DNA binds to the origin of replication. So let us go back and see here. So this particular site now is enacted upon by DNA helicase, an enzyme called DNA helicase. Now what does this DNA helicase does? Now, so this particular area, what do you see here, was actually the some of this particular region had this origin of replication. Now what happens is this enzyme called helicase, as the name suggests, it is a helicase. What it does, it unwinds the helix. So DNA is basically is in the helical form, right? It is wound on its own self. The two strands are wound on itself, right? And then there are these bases in between, right? So what it does is DNA helicase enzyme, this basically binds to this particular region called the OREC, and it breaks these hydrogen bonds between the base pairs. So you got like adenine and thymine, which are bound by like two hydrogen bonds, and we got G and C, which are interlinked with 3 hydrogen triple bonds, and then we have got like phosphodiester bonds here, and we got like a phosphodiester bond here, and similarly we got another nitrogenous base, and so this entire sequence is there. So what does this enzyme DNA helicase does? It breaks these hydrogen bonds, and when these hydrogen bonds are broken, we get 2 strands. Now these 2 strands, which were bound together by the hydrogen bonds, will now separate out something like this. So we got like these 2 strands, and they separate out like this, you see. And here we have the pairing. Here we have the pairing between the nitrogenous bases, but this particular region we do not have any pairing, and this particular structure that is found is known as the replication bubble. See, it appears like a bubble in the middle of the chain, and that's why it's called the replication bubble. Now we also have got like 2 ends here. One end is here and one end is here. If you look closely, this particular end, it looks like basically a fork, a spoon and a fork. So the fork that you might have seen have a lot of these prongs, but now this particular fork has only 2 prongs made out of 1 strand each. So because this particular structure, this structure here, it appears like a fork, we call this as a replication fork. So we've got a replication bubble, and then a replication bubble gives rise to 2 replication forks, one on this end and the other replication fork on the other end. So we've got like 2 replication forks here. I hope this much is clear to you how the process of initiation starts up. So the process of initiation is brought about by the protein called DNA, which if you don't remember is all right, but if you will have to remember that certain proteins are involved in identification of this OREC. And DNA helicase, it opens up or splits the strand, it tears apart the strand into 2 by breaking the hydrogen bonds, and the structure that appears like a bubble is called the replication bubble, and the 2 ends, one end is here and the other end is here, each of this end is called a replication fork. So the process of initiation generates 2 replication forks, and this is an active process, requires a lot of energy, right? So 2 replication forks have been formed. Now, because the 2 strands that have been separated apart have got complementary bases on them, right? So for example, here what do we have? We have got, let us assume let's draw a particular sequence, A, T, and let's say G and G and G, and let's say T, and that is all. Okay, so like we got 1, 2, 3, 4, 5, and 6. Let's say we got 6 bases here. On here, what will we have? We'll have complementary bases. So we'll have a T here, we'll have an A here, we'll have a C here, we'll have a C here, we'll have a C here, and we'll have an A here. So these nitrogenous bases which have been separated apart are still extremely in the unstable state. They still can pair up, right? Because there is this attraction between the 2. Hydrogen bonds can be formed even though they've been split apart, but they are still trying to attract each other, trying to form bonds back, the broken bonds back. So what happens, there are these particular proteins called the Ssb proteins, or single strand binding proteins, and what they do is, let us just magnify this image here. And so what it does is, we are just going to look at one particular replication fork here, one side of the replication fork. So what do we have here? We have this blue one is the helicase enzyme, and it is breaking these hydrogen bonds between nitrogenous bases. And the base pairs that have been separated apart, each base of the base pair is now bound to these particular proteins, and these are called Ssb proteins. What Ssb proteins do, they prevent these separated bases to pair up again, right? So they provide greater stability to the single split strands, two anti-parallel strands. They stabilize them, they prevent them from bonding back again, annealing again, right? So let us just go back. So I hope you've understood the function of the Ssb protein, single strand binding protein. Let us just enlarge it a bit and bring the slide just a little to the center. So far we've seen in this particular process initiation the role of different proteins and the different structures that are found in replication bubble and replication fork. Now let us go to the second stage, which is the process of elongation. Now see this plant, the two strands have been split apart. They've been split apart, and now two more strands must be formed. So if you remember the semi-conservative mode of replication, what do we have? We got like the two parental strands will be split apart, and two new strands, absolutely new strands, are formed on each of them. So this is what is about to happen, right? This is what is going to happen in just a little while, and this happens by the process called the process of elongation. Now elongation, what happens in elongation? Number one, nitrogenous bases. So what is required? Basically we require nitrogenous bases and we require energy to basically add up these bases. So if you go back to the previous slide, magnified it just a little bit and see. So let us just write a couple of letters here. Let us say, right, we have got an A here, and let us say this is T. So if a new strand has to be built here, or let us take this particular strand here, let us say this is T, and let us say this is A, and this is G, and this is C. So if a new strand has to be built out here, what do we need? See, what do we have here? We have a nucleotide here. We have another nucleotide here. These two nucleotides are joined to each other by a phosphate bond. So we got a nucleotide here. We have a phosphate bond here, right? So what do we need here? A complementary strand must be built here. So nucleotide must be paired to another nucleotide. So we need, so let us assume if this is T, we would require a nucleotide here, adenine nucleotide, and here we need a thymine nucleotide. Here we require a cytosine nucleotide. Here we require a guanine nucleotide, right? Now, so first thing that is required in elongation is, the first requirement are those of nitrogenous bases, and then these nitrogenous bases have to be added. A phosphodiester chain also has to be formed. See, if you go back to that slide. Now, not only these base pairs have to be connected to these bases, not only these bases, adenine not only has to be brought close to thymine to form a hydrogen bond, but also one more thing has to be done. It should also have a phosphate backbone connection with the next nucleotide, which will again form a hydrogen bond. So for the formation of this phosphodiester bond and for the formation of these hydrogen bonds, energy is also required. So for the process of elongation, we require nitrogenous bases, we also require energy. And where do we get all of this from? Now we don't necessarily use ATP here. What we rather use is, so there are these particular the substrate that are used that act both as a source of nitrogenous bases and as energy are called Dntps. And what do these stand for? They stand for Deoxyribonucleoside Triphosphate. So deoxyribonucleoside, what does that mean? It means a base, it means a sugar. So that's a nucleoside. Why do we call it deoxyribonucleoside? Because the sugar here is a deoxyribose sugar. So deoxyribonucleoside, but how many phosphates do we have? We've got like 3 phosphates attached to it. Now these particular Dntps, they serve as the source of nitrogenous bases and also serve as the source of energy. And how? Let us just have a look into this particular diagram here. So let us just magnify a bit and see a Dntp. So here in this particular image, we have got a guanine nucleoside triphosphate. So this is guanine nucleoside. If you only look at this particular part here, let us just draw. If you only look at this particular part here, this is a nucleoside. Right, it's a nucleoside. We have a base and we have a sugar. So this is a nucleoside. And how many phosphates do we have attached to this? We got like 1, 2, 3. So this is a guanine nucleoside triphosphate, right? And this particular Dntp, when it adds up to this particular chain, it forms a phosphodiester bond here. We'll just look into the details in just a little moment, right? It not only forms a it not only provides energy. These 3 phosphate molecules that are attached to each other. They attach to each other via these high energy bonds. When these bonds undergo hydrolysis, they release a large amount of energy, and that energy is used to connect this phosphate to this hydroxyl to form a phosphodiester linkage. And the two bases here are also brought together. Now we'll just have a look at them in just a little while in slightly greater detail. But at the particular moment, we only need to understand the sources, the basic requirements of the process of elongation. Now, the process of elongation requires nitrogenous base, and what provides the energy? Dntps, Deoxyribonucleoside Triphosphates. What else is required for the process of elongation? We require an enzyme called Dna Primase. We'll see why. We require an enzyme called Dna Polymerase, Topoisomerase, Dna Ligase, and Dna Polymerase, another kind of a polymerase. So there are different types of polymerase enzymes. We'll look at their functions in just a little while. But before we proceed any particular further, we need to understand the role and the conditions required for the functioning of one of the most important enzymes of the process. Now, elongation, the main enzyme in the process of elongation is Dna Polymerase. Now, I hope you remember, you know the process, you know what polymerization means. It is to connect same kind of things over and over again. It is to polymerize. Okay, polymer. You take similar units and connect them together, and this what have we found? We have found a polymer. So in one way, if you look closely, DNA is nothing but a polymer. DNA is a polymer of Dntps, or if we say nucleotides. DNA is a polymer of nucleotides. So there are these different nucleotides. So here we have got one nucleotide, here we've got another nucleotide, third nucleotide, so on and so forth. We've got a large number of nucleotides. So DNA is a polymer, and if a new DNA has to be formed, basically what are we saying? We are forming a polymer, right? So the synthesis of a new strand is polymerization, and the chief enzyme is called Dna Polymerase. Now, Dna Polymerase has got a certain set of conditions only under which it works. So Dna Polymerase, first of all, cannot work all by itself. Let's go back to this particular strand. Let me just clear this up and tell you what I mean. So now the chain has been split apart here. Okay, here the chain has been split apart, and forget about everything that is drawn. So for example, we have a Dna Polymerase here, forget about it, because this is slightly advanced. We'll just try to understand from the very beginning. Okay, so let me just draw. Let's say this is Dna Polymerase. Now the Dna Polymerase is around, and it cannot actually directly go to this particular part and start adding nucleotides here and forming phosphodiester bonds. It just cannot do it all by itself. What Dna Polymerase, what this particular enzyme Dna Polymerase it requires a pre-existing chain. It requires a little segment of DNA in order to start this process. It requires a pre-existing chain of base pairs already connected here. So let's go to a fresher space. Let me just go to the new fresh sheet and tell you what I mean by that. So let us say we got like these two separated strands, okay, and here like we got these base pairs. So let me say I've got these base pairs ATGC, ATGC, and CG. So for example, this is a base. This is one of the strands, and let us say this is a Dna Polymerase. So Dna Polymerase cannot act here. What it requires a small segment of already complemented tiny little strand here. So what it is looking out for? It is looking out for a segment. So it is looking out for something that has already paired with this, a tiny small segment. So it is looking out for a segment like this, and C and G and T, and they are connected to each other by the phosphodiester linkage. So this particular tiny little segment, which is required by Dna Polymerase to start functioning, is called a primer sequence. And a primer is not a DNA sequence in nature. It is basically a very small RNA fragment. So if this has to be an RNA fragment, what should we do here? We should not have a T here. Adenine, right? RNA does not contain adenine. What it contains instead is a uracil, right? So just to sum up, Dna Polymerase cannot start working on the separated strands by themselves. They require a tiny little segment called a primer, which must be already present complemented with the original strand. So it requires a slight bit of primer, and if the primer is present here, what Dna Polymerase does, it starts adding up bases from the next base. So it will start adding a particular base here, a particular base here, a specific base here, a specific base here, and the chain will be elongated. Now, how is this primer formed? What will form this tiny little primer? Certainly an RNA primer is needed for the beginning of the functioning of DNA polymerase, and this primer is formed by an enzyme called DNA Primase. Now let's get back to the process of elongation and see what is happening there. So here we have got this enzyme called DNA Primase. Have a look here. So we have got an enzyme called DNA Primase. What DNA Primase is doing? It is going to form these tiny DNA primers. And once this DNA primer is formed, DNA polymerase can actually start making a chain and elongating the newer strand. So let's just go back to our original slide. So DNA polymerase, the first thing that we learned about DNA polymerase is it will only work if a primer is present. What forms a primer? DNA primase, right? And what is basically the chemical nature of primer? It is a very small RNA segment complementary to the opened up or the nascent or the single DNA strand present in the replication bubble, right? So these are primers. Now also there is another characteristic of DNA polymerase, which is DNA polymerase can only synthesize a new strand in the 5' 3' direction. What do we mean by this? Now let us look at this particular diagram once again. All right, let's look at this particular image. Let's just bring this slightly in the center and let us see. So we got one strand here and we got one strand here. So this is the 5' 3' strand. Let us just write it down. So we got like, this is the 5' 3' strand. You see, this is the 5' end. So what do we have here? We have got the deoxyribose sugar, and we have got the deoxyribose sugar, and this is the third carbon here. So this is the first carbon, this is the second carbon, this is the third carbon. Here we got the fourth carbon, and this is the fifth carbon. To the fifth carbon, we've got a phosphate group that is attached, and to the third carbon, we again have a phosphate group, but which is attached to the fifth carbon of the next deoxyribose sugar, right? So now let us say in this particular strand, we have a strand here. Where can the Dntp be added? Dntp. So let us say this is the deoxyribonucleoside. What are the functions of Dntp? First of all, it will serve as a source of the nitrogenous base, the complementary nitrogenous base. C will pair up with G. So we got guanine nucleoside triphosphate here. And what is the function of these phosphates? They will provide energy, and they will also provide one phosphate for the formation of the backbone. Now this Dntp, where does it join? The phosphate is always bound to the 3' hydroxyl group. See, the phosphate cannot be added here because phosphate is already there. See, the 5' end already has a phosphate. Can we add this phosphate here? We cannot, right? But where is the point where these phosphates can be added up? They can only be added to this 3' end. So new bases, new Dntps are always added on the 3' end of the DNA strand. So we got like these two strands. One strand is here, one strand is here. So this Dntp is added out here. Two phosphates are used up to produce energy, and they are released outside as two phosphate groups. They are no more required. Once the bonds are broken, they are released outside. So two phosphate groups are released, and then we got this one phosphate group. It forms a bond with here with the 3 carbon. Certainly it's also a dehydration reaction, so also we get a certain amount of water outside, and so we have a bond here, and this is the phosphodiester linkage, which has connected basically two deoxyribose sugars and a guanine, right? So what is the direction of synthesis of DNA? This is very important. You must remember the direction of synthesis is always from the 5' end to the 3' end. So this is the direction of formation of DNA or the synthesis of DNA. And why is this so? Because the phosphate group can only be added to the 3' end, hydroxyl end of the deoxyribose sugar. Now let us have a look at the other end. And on this particular end, where can we add a Dntp? Can we add a Dntp here? Dntp here. So the Dntp cannot be added here because it already contains a phosphate. The Dntp will always be added onto the 3' end. So you see, when new strands are synthesized, one strand will be synthesized in this direction and the other new strand will be synthesized in the other direction. But most important for you to know is that the DNA, the newer strand that is synthesized from the 5' direction to the 3' direction, and we'll see to it in slight greater details in just a little while. But at the moment we need to understand the properties of DNA polymerase. So first property of DNA polymerase is that it can work in only one particular direction, and it requires a primer for its functioning, right? It can only work in one direction. It can only add nucleotides to 3' end. DNA is always synthesized in 5' to 3' direction. Now, so far we've done DNA primase, we've done DNA polymerase, and in particular we've done DNA polymerase 3, but you don't need to remember this, just remember DNA polymerase. Now, what happens is, let us just go back a little bit more. Now we are going to go to the function of DNA topoisomerase. Let's go to this particular slide, and this is our next slide. So far what we've seen is, let us look at the first step. What has happened so far is helicase has unwound the parental strands, and single strand binding proteins were bound to it. And then the third step is, now we've got the process of replication going. So now we got we have two strands here. Let us demagnify it just a little bit and get back to understanding what is happening. So we have like two strands here. Okay, so I want you to look at this particular strand. Okay, let us look at this dark blue strand here on the top, this one, and the orientation of this parent strand. So we got two parent strands now. We got one, this is one parent strand, and this is the other parent strand, and both these strands have been separate here. Say these are two separate strands here. So these are two parent strands. Now the parent strand, the two parent strands have got two different orientations. So this particular strand is a 3', 5' parent strand. So this is a 3', 5' parent strand. So what happens here at the beginning? So let us say when this was just opened up, when the replication fork has just begun, the replication fork would have been somewhere here, and RNA primer would have been added up here. So let us say we have an RNA primer here. I want you people to please pay attention right here at this particular point. So at the beginning, so we have a replication fork here now. The replication fork has basically moved, but at some point of time in the past, the replication fork would have been here. So when the replication fork was formed, this is the 3' end, a little RNA primase was added up, a little primer was added, which is basically a small segment of RNA. And because this is the RNA, this is the new complementary strand, this is the 5' end of the RNA primer, and this is the 3' end, right? So the DNA polymerase, this particular enzyme here, actually binds to the 3' end. It binds here, see, at the 3' end, and it starts adding up Dntps. So one Dntp is added, another Dntp is added, and then a phosphodiester linkage is formed between the two, and then another, and this enzyme keeps moving forward, right? And then another base is added, another base is added. So we see that the direction, because polymerase, DNA polymerase moves in the 5' to the 3' direction, and so this is the 5' end of the RNA primer and 3' end of the RNA primer. So the polymerization is taking place in this particular direction. New nucleotides are being added. Who's adding them? DNA polymerase. And the new nucleotides that have been added are also connected to each other via a phosphodiester bond. And who's creating the phosphodiester bond? Again, the DNA polymerase enzyme, right? So the synthesis is happening continuously on one of the parent strand, and the DNA polymerase moves forward, keeps doing the same thing, and it reaches here. So you see on one of the parent strands, which parent strand? On 3', 5' parent strand, the process of DNA synthesis or DNA polymerization is continuous. Here, the DNA strand is being made continuously without any particular break. Now, why is this happening? Because the 3' end of the primer is here, right? As a result, because of the complementary, because the nature of complementarity. And we have a 3' end, and that's why all continuously we have a new DNA daughter strand that is being formed here. But if you look at the other strand, let us now look at the other parent strand. And what is the other parent strand is? The orientation of other parent strand is 5' to 3'. Let us look at the other parent strand. So the other parent strand is 5' to 3'. Now when this chain opens up, let us assume it opened up right here. When this chain had opened up, it will also get an RNA primer attached to it. Now the RNA primer, so let us say this, in order to understand this, let us make a newer diagram here. Let me just erase this. So this has been erased. So let us say now, so this is one parent strand, and let us say this is the other parent strand. And here we have got the, so this is the 5 prime, 3 prime, and the 3 prime and 5 prime. On the 3 prime 5 prime we've got a continuous strand that we just understood. We have a continuous strand. And how many RNA primers do we have? We just had only one RNA primer here. But when the other strand, when that opens up, so let us not assume that so much has opened up. Let us just shorten it a little, and let us assume that the opening is just this much. So this is opened up, and this is the 5 prime end, and this is the 3 prime end. So on this particular strand, 5 prime 3 prime strand, as the strand just opens up, let me just make it slightly longer. Okay, so now the strand has opened up, and an RNA primer will be added up. Now because this is the 5 prime end, we will have a primer with a 3 prime end here and a 5 prime end here, right? I hope you're getting this point. Why do we have so? So in the other parent strand, this is the 5 prime end, and if an RNA primer has to be, if a primer is added, let us say this is the primer with a couple of bases. If the primer is added to this 5 prime end, only a complementary primer can be added, because the strands are anti-parallel, they are anti in nature to each other, orientation is different. Here we've got the 5 prime end of the deoxyribose sugar, and on this we have got the 3 prime end of the deoxyribose sugar. So here we have the 3 prime end, and we have the 5 prime end. Now just think about this. The chain is continuously opening. We have got a helicase enzyme which is completely opening up the chain and elongating the fork. So here what do we have? We have got a strand right here. Let's use the original color. So the strand is continuously opening, and on this particular strand we have got a primer from so we have got a 3 prime a 5 prime and 3 prime primer, and we got a new daughter strand forming. But here we have a problem. We've got a primer here, and if DNA polymerase comes here, it will have to work somewhere here. And where does it work? It only adds on the 3 prime end. But on the 3 prime end, at the moment we have nothing. So, but the chain has elongated, right? So now let us assume that the chain has elongated quite a bit more. Let us make it a little more long. Let us say this is the chain now. The initial primer is here, and let's say this is the primer. This is the 3 prime end of the primer. This is the 5 prime end of the primer. This is the 5 prime end of the parent chain. Now DNA polymerase cannot act here because it has nothing to add on to this side. So the chain opens up, and when it is opening, it finds another particular region, and then another RNA primer is added here. So we see what do we have here. Now this is the primer. Again we'll have a 3 prime end of the primer and 5 prime end of the primer. And now see, one of the polymerases couldn't start the working here, but now another polymerase enzyme which is present around can work here. It actually can work here because it has got this template that can be copied. So DNA polymerase starts working on the 3 prime end, and it starts adding, let us use a green, it starts adding more bases, and you see we got a new strand that has come up, and it reaches here, and you see we have got a small fragment of DNA daughter strand. We got a small fragment, and once it reaches the next primer, DNA polymerase cannot connect these two. It does not have the ability to connect this small fragment of daughter DNA to the primer. It cannot do that. And meanwhile this is happening, the strand is still opening up more. This strand is still splitting up more. So as this helicase is still unwinding it more. So let us unwind it a little more and see what is happening. So we got more unwinding happening, and let us say we got the helicase somewhere here, and you got like lots of base pairs here. So in the upper strand, the original strand, we have a continuous chain of DNA that is being formed. But here what do we have? We got more primers that are being added. So we got a primer here now, for which we have a 3 prime end, and we have a 5 prime end here, and a new strand is synthesized here, and that is only a fragment of the newer strand. So you see on one of the strands, which strand are we talking about? We are talking about the strand with the orientation of 5' to 3' parent strand. So if you look at this strand, I'm talking about this strand, 5' to 3'. If you look at this particular parent strand, on this particular strand, the synthesis of DNA is not continuous. It is the synthesis of DNA or the copying of DNA on 5', 3' parent strand is discontinuous. And because it is discontinuous, it is lagging. The process of DNA synthesis is kind of slower because we got primers here, we got interruption, we got primers here. So the process of formation of newer DNA on this strand is discontinuous, and it is also slow, which means it is lagging this particular strand. This newly synthesized discontinuous broken up strand is also called the lagging strand. So this daughter strand, which is synthesized on the 5', 3' parent strand, is called the lagging strand. And the strand which is synthesized on the 3', 5' parent strand, 3', 5' parent strand is called the continuous strand, or because the synthesis of DNA is continuous there, that strand is called the leading strand because it is leading. It is working faster. It is more continuous, it is more mature than the other strand. So this is also called the leading strand. Now remember, leading strand and lagging strands are the name of the two daughter strands. Now these fragments that we see here, the fragments that start from the RNA primer and with a short segment of complementary DNA daughter strand, these segments here were discovered by a Japanese scientist called Okazaki, and in his honor we call these tiny little fragments of DNA present in the lagging strand as Okazaki fragments. Right, so all of this is a part of the process of elongation. Now you might wonder, how are these strands then connected to each other? What happens to this RNA? Now there's this another enzyme. Now I've told you already that there are different types of polymerases, different types of topoisomerases. So far the DNA polymerase that you saw, so this polymerase enzyme that was making newer strands here on the leading strand, on the lagging strand, is called Dna Polymerase 3. This is called Dna Pol 3. Now there is another enzyme called Dna Polymerase 1, and that enzyme has the function of basically breaking down all of these RNA primers and replacing them with appropriate bases, nitrogenous bases, complementary bases. So that is what the DNA polymerase does. It basically removes these RNA primers and then replaces them with the DNA. But you must remember that in spite of removing the primer sequence and replacing it with the DNA sequence, there still is this gap in between the two fragments. Between the Okazaki fragment, there is this gap. And how is this gap filled? Now there are two adjacent bases only. This particular gap, because see, there is a gap and that has to be welded. How can a welding be brought about? How can these two joints be connected? These two joints can be connected by a phosphodiester bond. And this joining of the two Okazaki fragments, to join, there is another word, there is a word used for joining which is called ligation. Ligation is the same as joining or welding. So the two Okazaki fragments, after DNA polymerase 1 has removed the primer and replaced the primer with the complementary DNA strand, the enzyme called ligase, there is another enzyme called ligase which comes and connects these two broken out fragments. And so what do we have here? So this is, let us just look at this particular representation here as a summary. So we got a continuous strand, continuous replication, and we got a leading strand here, and then we got lagging strands here, and the lagging strands are acted upon by DNA polymerase by removing the primers are removed by DNA polymerase 1, and the segments, the newer formed Okazaki fragments, the gap between the two Okazaki fragments are then joined together by DNA ligase, and hence now we are getting continuous strands or newer joined strands on the lagging strand also. So the joining process, basically the processes involved on the lagging strand are a little more complex compared to the processes that are going on the leading strand. So where have we reached? So we've just finished the process of elongation. So this is elongation. So far, let us just summarize very rapidly. So we've seen the function of DNA primase to form the primer, DNA polymerase 3. Now we'll have to look at the function of another enzyme which is called topoisomerase. So now we need a newer slide for this. Let me just erase this. So these are the two strands, let us say. Okay, let me just redraw it. So we got like the two wound up strands, and then we got the replication bubble here. We got the replication bubble here, and then we got the two winding strands. And the replication forks, one replication fork is moving in this direction, one is moving in this direction, and we got like one helicase enzyme here, and we got one helicase enzyme, and the helicase enzyme is basically pushing and ripping the two strands apart. So one strand is being pulled this way, one strand is being pulled this way. Now if we continuously, if the helicase is acting in this particular manner, if the two strands are pulled apart by helicase, at some point there will be a lot of tension between in the strand, in the complementary strands in front of helicase. There will be development of stress or tension. Why? Because this particular coiling, so these are, for example, these are the coils. Let's say 1, 2, 3. We got like 1, 2, 3, 4. We got like 5 coils in this particular segment. But as the DNA helicase will push this, as it is making its way forward, there will be a lot of coiling, and you will find that they will come closer to each other, and there will be a lot of stress, and this particular stress can actually break or damage the DNA. So in order to remove, so basically this is called this is a topological stress caused by supercoiling. And why is supercoiling happening? Because the helicase is ripping apart the two parallel anti-parallel strands and then creating tension in the paired up strands in front of it. Right. So there is this another enzyme which is called topoisomerase, and we'll have to look at it in a diagram, and let's go back to this particular diagram here. So there is this enzyme right here in the image. This is called topoisomerase, and what this enzyme does, it releases this tension that is caused in the strand in the front. And how does it do it? It simply makes a tiny little cut and then allows the respinning of the strands and then connects them back again. So what do I mean when I say this is, for example, we got these particular strands. Let's just rub what is not necessary here. So okay, let's just rub everything and draw it once again. So let us say this was the original strand, and now this is the strand in which we have a lot of tension that has developed. So what the topoisomerase does, it goes out here and makes a cut, or probably let us say it goes out here and makes a cut. And once it cuts it out, what will happen? These particular strands that have been superly coiled will unspin and will become uncoiled, as you can see here, and it becomes uncoiled like this. So say in this particular section we have got the uncoiling and the stress has been released. So there's this enzyme called topoisomerase which just does this, right? So we've seen the function of all the enzymes here: DNA primase, DNA polymerase, topoisomerase, DNA ligase, and yes, there is another enzyme, DNA polymerase 1, which we saw, and the function of DNA polymerase 1 is to remove the primer, replace the primer with the original with the actual complementary DNA strand, right? And the function of ligase is to connect the two Okazaki fragments together. Now this entire process of polymerization or elongation of the chain takes place at an incredible speed, the speed of around 2000 base pairs per second. This is how fast the process of addition of these base pairs is going on. And what is really amazing is that there is only one mistake made in 10 to the power of 10 base pairs. For every 10 to the power of 10 base pairs that are added up on both the strands, there is only one mistake, which again happens because of certain repair capabilities or proofreading capabilities of these enzymes, which you do not have to understand at your level. Now let us go to the last part of DNA replication, which is called termination. Now termination, as we studied at the beginning, the process of termination takes place at the ter site. So if you remember, here we have the original circular DNA of the E. coli, and we had started from origin ORI-C. This is the place from where the replication bubble formed, and we've got two replication forks. We have one moving in this direction, we've got one moving in this particular direction, right? Now the two replication forks are going to move and move and move till they reach a particular segment or sequence called the ter site or the termination region, and this is what it looks like. Now we got two replication forks, and we got now one new strand here, daughter strand here, we got one new daughter strand here, and we got two new DNAs here. Now when both the replication forks, when they reach, so I want you to look at this particular image here. So here we have got one replication fork exactly here, we got one replication fork exactly here, and then they reach this particular region called the TER region. Once this TER region is reached, there are these proteins called the Ssb proteins which identify this. So the Ssb proteins identify the termination region. The two replicating forks, when they reach the ter region, they actually merge into each other. So this particular end of the replication fork and this replication fork, they will meet at this particular point, and then they will separate out from each other by forming two exact complementary sequences. And now what we have here in this particular image, you can see we got two strands, two new daughter strands. We had one parent strand, replication started, two replication forks were made. Eventually at the ter region, TER region, there were Ssb proteins, and these are used for the identification of the ter region. Once the termination region is reached and replication forks identified, the two replication forks, they meet up, and two new daughter strands are freed from each other. But if you look at this, what we are getting is both these strands are still linked to each other like two rings. And one more enzyme called Topoisomerase 2. Now this is a bit of complication, that there are large number of topoisomerases, there are wide varieties of polymerases. So this is another kind of a topoisomerase which makes a cut in one of these strands and frees this strand and rejoins this strand, and then we got two new daughter strands. So this is the process of DNA replication in fair details.